Hi, I'm John Deck from the University of California, Berkeley. And so I'm talking about Biocode Commons, which is a name that we've given to essentially an informatics stack for genomic observatories. So what is this? We've coined this as resources for genomic observations from collections to analysis and publication. So the essential approach here is looking at three main areas: tools, standards, and data. And we've heard a lot of great talks in the last couple days looking at some tools, all the informatics type talks, or looking at data or looking at standards. But generally what happens is that we get stuck in one of these realms. We're looking at purely a tool development, or you're looking at purely in terms of standards, or you're looking purely in terms of data. So the challenge here with Biocode Commons is looking at everything that sits in between these areas. So Biocode Commons is not meant to be a specific tool or a standard or even store any data. But what we want to do is we want to find those areas that lie between this. How can we effect better cross-communication between these different areas? So what I'm going to show you is a little bit some ideas that we have for how maybe we can approach this. And then there's some discussion questions, which I'll just show you briefly at the end. And then we have this, a breakout group this afternoon where we can really delve into this further. So I worked for several years developing the informatics for the Maria Biocode project, and Neil showed this slide yesterday. And really what this shows is that we have a lot of different types of data from the Maria Biocode project. And I think this is a pretty good, it's kind of like Maria as a small island in the middle Pacific. And we saw where it sits. I think it acts as a good microcosm for Biocode Commons because we worked with many different natural history museums. We worked with many different types of data. And we tried not to develop one single monolithic system. We tried to, recognizing that people who are collecting specimens and tissue samples and sending samples off to different laboratories, that we looked at it purely as a network. We couldn't develop a single system. So all of these things are interacting across domains and are distributed. So just to characterize the landscape, we're dealing with distributed data. There's a lot of emergent technologies, and there's multiple domains. And it's actually, I ran across this picture of the Star Trek utility belt, and a lot of the tools now really do look remarkably like the Star Trek tools, which I think is just really awesome. So this slide is not meant to be a comprehensive view of all of the standards or all of the networks, but it's just meant to convey, the environment that we're working in. So we're out in the field, we're collecting data there, we're working in the lab, we're doing analysis. How do those different areas, oftentimes there's databases purely for capturing field data, or oftentimes there's a lot of databases for capturing LIMS data, or analysis toolkits. How do they work together and how do they integrate by adopting different standards and workflows? So there's been a couple talks on workflow approaches for connecting data and standards, and really this is actually like tool-to-tool interaction, I think. And I don't myself personally have a lot of experience with that, so I think the BioKepler folks and the BioVal folks did a good job of describing what they're about. But I think that working with the workflow packages could be part of the Biocode Commons. I'll talk a little bit more about linked data approaches for connecting data and standards. That's just something I have a little bit more experience with. So the idea here is that by adopting standard terms, you'll notice that up in the right there that there's a URL that points to a particular term, which we call an event. And so the idea is that we can apply this URL to some type of activity or some type of object. So we're very clear about what it is that we're collecting. So this is important when you get into, if you're looking in one particular domain, everybody knows what you're talking about. You just use natural language and you call it this or whatever, and you don't have to be clear. But when you're doing cross-domain work, it becomes more and more imperative that we're very clear about those things. The next step here in working with linked data is that you assign an identifier to these terms. So using identifiers and globally unique identifiers is an important step. You can assign properties to these identifiers as well. You can link the identifiers together, and then you can publish them. That's essentially what linked data approach is. If we look at identifiers, a general, these are, I love identifiers because I think they underlie so much of the data when we're looking at distributed data and connecting different data sets across domains together. Identifiers are important. Persistence is a big problem, but some of these, we've been working on this recently. Solutions, DOIs have been becoming increasingly common. Easy IDs is a technology that California Digital Library is putting together, kind of making it easier to develop really good identifiers and make it easy to apply to things. Many people have been using Pearls. Assigning identifiers at the source is a very big challenge. And I think the closer you can assign an identifier to the actual point of collection or adoption or whatever you're doing, the better off you are. Oftentimes, the challenge in assigning a globally unique, persistent identifier to something that happens farther down the value chain. So we see these disconnects in data. You see a spreadsheet that's being collected in the field, and someone puts on a temporary identifier, they load it to one system, and then they load it to another system, and then they assign the identifier off in the first system here, and then they assign another identifier in the second system here. So we have duplicate identifiers. So these synonymies here are a big issue. And another issue is just a very simple thing, is that the semantic web and linked data requires URIs, but many standards don't require URIs for identifiers. So simple solutions here, for instance, just promoting the use of URIs for identifiers. There are some other solutions which, if you really get into it, you can talk about embedding UUIDs with other types of identifiers. Another thing that we've been working on a lot in the last few years is just clarifying terms and just realizing that a lot of our vocabularies really, I think, could use some help in terms of looking at, what exactly do we mean by something? And so the top example, occurrence, is a word that is used in the Darwin core vocabulary. It can mean simultaneously an observation of something in nature, so there's bird watchers up there, or it can also mean a specimen sitting in a drawer that has been accessioned into a museum. So, calling them both occurrence, that might be a little bit unclear, misleading. The other examples, there's many different things, it's like what you call a sample or a specimen, an individual or an aggregation may oftentimes become incredibly confusing when you're talking about different things. So the example of the termite here, are you looking at if you take the microbes out of the gut of the termite, is it still functionally a termite? I don't know. Or if you call that the sample, how do you differentiate the microbes within the gut and the termite itself? So same thing, looking at a jar of, I'm not sure what those are now, chitins or something that, so what is it, what is the relationship of those things that are sitting in the jar? Are they all taxonomically homogeneous, where they collected at the same spot? How do we break those out? So kind of another activity of Biocode Commons is purely just connecting developers, getting folks together and developing ways that we can facilitate communication between different projects. So here are a couple photos of some projects. We had a workshop in February where we got some folks that were experts in Darwin Core and mixes together and talking about how can we integrate Darwin Core and mixes and the different challenges there. We had Semantics of Biodiversity workshop with Barry Smith in May at the University of Kansas. And that was very valuable for folks who were wanting to look at just, again, term clarification in the Darwin Core and how different things may relate to each other using the basic formal ontology as an upper level umbrella and trying to help sort out these different concepts. So we're going to have, there's a few different breakouts this week that are related to Biocode Commons, and so I'm going to go over what these are. So there's immediately following lunch in Lecture Room C, we're going to have this, the first Biocode Commons breakout. We're going to talk about software, what kinds of software are relevant. We're going to talk about web services. You know, do we want a coordinated stack of web services? Is this needed? What are the things that are really holding people up in implementing and making great use of web services right now? How could we make them, you know, more used? What steps are needed to integrate workflows? In terms of data, just reiterating that Biocode Commons is not a data repository, but it's really how do we connect the different data repositories that you know, we've heard a lot about data repositories and different people that are handling data here. How do we get them together? So what kinds of data access permissions? You know, are these all open access, private access? Do we need to enumerate the types? How do we enumerate them? Who can we work with in terms of helping to solve, even just clarifying the different types of access permissions that could be possible? In terms of standards, you know, what are some strategies for global unique identifiers? You know, what kinds of standardized lists are important in this domain, and what ontologies are we using, the minimum information standards that are out there, biosharing catalog. Susanna's, you know, graciously said that we could use the biosharing catalog and she can help filter different types of content for looking at what is directly pertinent for genomic observatories. Later on this afternoon, there's going to be a governance session as well, which is, I think, actually a very crucial component here. How do we set this up? Who should be able to contribute? You know, how are we going to store the software tools and source code? Intellectual property rights, funding, who makes decision, licensing. And I think that's Steve Stones-Havas from Biomatters is going to be leading that session. And then later this week on Wednesday afternoon, we're going to have a hackathon where we're going to look at, it's a continuation of the Semantics of Biodiversity workshop we had in May. We're going to look at a biocollections ontology, and we're going to be using Protégé to help to model, to pick up where we left off in May. And we're going to be developing this biocollections ontology and also working on a ontology for Ocean Sampling Day. And then on Friday morning, there's another session led by Renzo, who's going to be doing, and in conjunction with MicroB3, the field sampling protocols and standards. I didn't mention on this slide that I think it's Friday at 1 o'clock, right? That we're going to be doing the wrap up. Yeah, so we're going to be getting the two hackathons are going to be getting together.
So field sampling protocol standards is 9 to 12, wrap up is 12 to 1, and
okay.
1 is...
Okay, thank you. And so 9 to 12 is the field sampling protocols, 12 to 1 is the wrap up, and 1 o'clock we're done. And that's it. Thanks.
Thank you.